my name is Rollin Leavitt, and thank you all for coming to my TED Talk. And I want to start out with this question. Who likes inherited diseases? Woo! No one? All right, then good. You're not going to be offended by my presentation. I started out my presentation looking at how we can actually combat inherited disease and what some of the methods of doing this are. The reason behind this is that inherited diseases are different than any other disease. Unlike an bacterial infection, which can be cured by a round of antibiotics, or a broken arm, which can be cured by a splint, there is no cure to an inherited disease. The closest we can do is try to treat the symptoms. However, this comes with its own problems. Immunosuppressants, which are meant to help cure autoimmune diseases, can leave the victim suffering from or being hospitalized from a simple cold. Because of this, we want to treat the cause of these inherited diseases as close to their origination as possible. The idea behind an inherited disease is that you have some sort of genetic mutation which leads to symptoms, and then the majority of treatment is focused on those symptoms. However, there's been a movement to move towards treating mutations before they cause symptoms, or trying to figure out how mutations lead to symptoms in order to figure out new ways of treating it. This question is really important in modern treatment of inherited diseases, how mutation leads to symptoms. One inherited disease which is at the forefront of this conversation is Crohn's disease. Crohn's disease is a complex inflammatory disorder which is characterized by inflammation and ulceration in the intestines, hypersensitivity to bacterial infections, elevated levels of microflora, so elevated levels of bacteria in the intestines, and then overactivation of certain transcription factors which help to control which genes are activated by your cells. Crohn's disease is interesting because there is no one-to-one cause between mutation and disease. However, we do see some genetic association, and so it's really important to look at which symptoms are elevated and caused by these genetic mutations. The two I want to focus on are elevated levels of microflora and overactivity of inflammatory gene regulators. That's because both of these symptoms have their root cause in mutations of this bacterial receptor protein called nucleotide binding and oligomerization domain containing protein 2, or NOD2 for short. NOD2's main purpose in cells is to detect bacteria. And so it is in the cytoplasm of cells, so it is searching for pathogenic bacteria that enter into cells and try to hijack their systems. Where it would be easy to look at how NOD2 affects one or two cellular systems, in the case of Crohn's disease, there are multiple cells which use NOD2, and so there's a little less clarity as to which process is abrogated by NOD2 mutation. The two main cell types which contain NOD2 in the intestines are called Paneth cells, which are basically specialized skin cells of your intestines, and then macrophages, which are immune cells which are looking to search out and destroy bacteria, like this one's doing. There are two theories on how the disease progresses and is caused by NOD2 based on these two types of cells. These two theories are called the defense and deficiency theory and the unregulated nuclear factor kappa B theory. For the majority of the history of NOD2 and its relation to Crohn's disease, it has been suggested that the defense and deficiency theory has been the dominant cause of disease. But there's some problems with that. So just kind of to quickly run through what this is, Paneth cells have a specialized job of creating these proteins called defenses, which, like the name suggests, defends the body from bacteria. Their job is to kill bacteria and keep the amount of bacteria in the intestines in check so that it doesn't overwhelm the cells in the body. NOD2 is the first step in the creation of defenses. It is what Paneth cells use to say, hey, there's bacteria here, I need to activate the defense system. And when NOD2 is mutated, this defensin production goes away, which causes unmitigated growth of microflora in the intestines, and the overgrown microflora is seen as a symptom of Crohn's disease. This has been the dominant theory of how NOD2 mutation leads to Crohn's disease for many years. However, recent studies have shown that only a single common mutation in NOD2 leads to a dramatic decrease in defensins. In fact, the majority of NOD2 mutations do not lead, while they do lead to some deficiency in defensin, they don't lead to enough to create a problem. Where they may be slightly larger bacteria growth, it's not enough to cause a problem. So this leads to the idea that defensin deficiency is not a cause of disease, but a symptom. Within this in mind, there is a new and modern theory relating NOD2 to Crohn's disease. And this theory focuses on NOD2's role in macrophages. So, really quickly, I want to describe what NOD2 is used for in these macrophages. NOD2 is the receptor protein which looks for bacteria. When it finds these bacteria, it causes a series of chemical reactions inside the cell to happen, which releases this protein called a transcription factor into the nucleus. The transcription factor then binds to specific parts of the DNA and says, hey, come code this gene. Transcription factors cause two types of proteins to be created by cells. Cytokines, which then leave the cell and communicate what's going on to other cells, and regulators, which stay inside the cell and help to affect processes in the cell. The modern theory of unregulated nuclear factor kappa B and NOD2's relation to Crohn's disease suggests that there's an overactivation of transcription factors, which causes, instead of a quiet conversation between your immune cells, a screaming match, and thus problems. The transcription factor that NOD2 uses to activate genes is called, or I guess before I should explain, there are two types of bacterial receptors that your macrophage cells use to look for bacteria. NOD2 cells, or NOD2 proteins and TLR4 proteins, which are toll-like receptors. In a normal cell, these two proteins communicate with each other to coordinate an immune response, because you don't want to over-respond to a little infection or under-respond to a big infection. So you have two receptor systems which are communicating to give you the right amount of inflammation. When either one of these are stimulated on their own, they cause huge amounts of inflammation. However, TLR4 has been shown to cause much more inflammation on its own than NOD2 on its own. Which is kind of weird, because both of these proteins use the same transcription factor. This transcription factor, the protein which moves into the nucleus and controls gene transcription, is called nuclear factor kappa B. Nuclear factor kappa B is a class of 5 subunit proteins, and the functional group is 2 of these bound together. Where NOD2 binds to the DNA and how it affects the DNA is completely dependent on the combination of these subunit proteins. The subunits themselves are divided into 2 categories based on their effects. The REL subunits have a pro-inflammatory effect on gene transcription, whereas the NFKB subunits, P50 and P52, have an anti-inflammatory effect. Depending on how these subunits are combined, you can see different levels of inflammation and different binding sites to DNA. As you can see here, a homodimer of REL-A would be super inflammatory, a heterodimer of REL-A and P50 would be somewhat inflammatory, and then a homodimer of P50 might actually be anti-inflammatory. Now I know what you're asking yourself. Rollin, if we know so much about these transcription factors, why can't we just block these sites and bring everyone back to normal at the root cause of these problems? Unfortunately, because of the different combinations and different activating factors of nuclear factor kappa B, there are a lot of binding sites for nuclear factor kappa B in the genome. And I mean a lot of binding sites. But I'm going to point out 2 in specific. A cytokine, which is the types of proteins that are released from the cell, called tumor necrosis factor alpha, and then a regulator, the types of proteins which are kept inside the cell, called interferon regulatory factor number 4. I'll come back to IRF4 a little later, but just remember that it is created by nuclear factor kappa B. For now, I'm going to focus on tumor necrosis factor alpha because it plays a really big role in Crohn's disease. Because tumor necrosis factor alpha causes apoptosis. Woo! Cell death. This is a really interesting process because it's at the root of ulceration and inflammation in Crohn's disease. Not only does it cause an elevated immune response, but it causes cells to actually die, which is a big problem if you have way too much floating around. In fact, there are multiple treatments for Crohn's disease which are focused on blocking tumor necrosis factor alpha, preventing it from interacting with cells. So we get this idea that when NOD2 goes wrong, there's too much tumor necrosis factor alpha. And there's an idea for this. Like I said before, TLR4 and NOD2 coordinate a response, and so they give a regulated amount of nuclear factor kappa B, which creates a moderate amount of tumor necrosis factor alpha and controlled cell death. Enough to make it work well. When NOD2 is mutated and it's no longer working properly, instead of seeing less nuclear factor kappa B, which we would expect because one of the activators is gone, we see more, and a lot more, and thus unmitigated cell death. That's kind of counterintuitive, right? How would getting rid of something that activates nuclear factor kappa B cause more nuclear factor kappa B to be made? And the answer is in the cellular process of NOD2 signaling. So I'm going to kind of take you guys through the process of NOD2 signaling from activation when it first finds the bacteria to when it creates the cytokines to be released from the cell. NOD2 is in a auto-inhibited form in the cytoplasm, so it's just kind of floating around, just chilling until it finds a bacteria part. When it finds a bacteria part, it binds to this protein called receptor interacting protein 2, and this is the beginning of the activation of nuclear factor kappa B. RIP2 is then polyubiquitinated, which basically means there are a bunch of little proteins added to it called ubiquitin. This is important because it is needed for the key to unlock nuclear factor kappa B. This key is called TAC1, and it allows nuclear factor kappa B's inhibitory unit to be gotten rid of, and then the transcription factor to enter into the nucleus and cause cytokines to be released. This exact same process is used by TLR4 to activate nuclear factor kappa B. However, there's one key difference. Remember that protein I was talking to you about before? Interferon regulatory factor 4?
Yep.
IRF. IRF. All right. This protein is really interesting because it inhibits TLR4 response, because it prevents, it occupies the binding sites for ubiquitin. So of course, if TLR4 can't be ubiquitinated, it can't activate nuclear factor kappa B. What we see in people with Crohn's disease, where NOD2 is mutated, is that there's not enough IRF4 produced, which causes this TLR4 pathway to run as much as it wants unregulated. I know this is complicated, so to put it simply, there's a balance of activity between the cells. NOD2 creates IRF4, TLR4 creates tumor necrosis factor alpha. When NOD2 is mutated, not enough interferon regulatory factor 4 is made, which allows TLR4 to make too much tumor necrosis factor alpha. This leads to cell death, to inflammation, and to the hypersensitivity seen in Crohn's disease. Or at least we think. There's a problem with this theory. And that problem is right here. The exact same process is used to activate nuclear factor kappa B by NOD2 and TLR4. And so we have no idea why we see different responses from NOD2 activated NFKB and TLR4 activated NFKB. In order for this theory to move forward into the clinical stage where treatments might be developed from it, we need to explain how this works. Remember those nuclear factor kappa B subunits I was telling you about? The REL subunits are inflammatory, NFKB subunits are anti-inflammatory. Well, it turns out the anti-inflammatory subunits have binding sites for interferon regulatory factor 4. And the pro-inflammatory subunits have binding sites for tumor necrosis factor alpha. From this, it seems like toll-like receptor 4 has a preference for these proteins, and NOD2 has a preference for these anti-inflammatory proteins. Which brings me to my question. Do the NOD2 and TLR4 bacterial receptors have differing transcription factor dimer preferences, and can this preference account for the observed inhibitory interaction between these two proteins? To test this, I went into the lab at Fort Lewis College, and I got down to work. So I got my microscope, my spectroscope, my stethoscope, and some scope, and I went to work. Through a few experiments called an ELISA experiment and a Western blot experiment, I was actually able to correlate amounts of tumor necrosis factor alpha produced by cells which are stimulated with the NOD2 ligand or the toll-like receptor 4 ligand, and I was able to look at the amounts of transcription factors that move into the nucleus when they're stimulated. This data shows the concentrations of tumor necrosis factor alpha produced by cells after they've been stimulated. As you can see, the blue lines, the ones that were stimulated, or the TLR4 stimulated cells, have a much higher rate of tumor necrosis factor alpha production than the ones stimulated with MDP, the NOD2 ligand. And this data actually correlates with my Western blot data. As you can see here, this is a Western blot data analysis, and so basically the brightness of the band shows how much of the protein is there. This is a nuclear isolation blot. So what I did is I stimulated the cells, made all the transcription factors activate and move into the nucleus, then I ripped out the nuclei, mushed them up, I mixed them up with a bunch of glowy stuff, and then I put it on paper. And as you can see here, there is much less REL-A in the nuclei of MDP-stimulated cells, NOD2 activated cells, than TLR4 activated cells. And so, does this mean that NOD2 and TLR4 have transcriptional factor preferences, and this accounts for their inhibitory interaction? Probably. At this point, I'm still in the middle of my testing, and so I'm going to be going back into the lab over the next month and a half, and I'm going to be testing other subunits. And if I see the same interaction with C-REL and REL-B, the other REL proteins, being preferred by TLR4, and if I see the NFKB proteins being preferred by NOD2, then I will have proved my findings. As always, data in and of itself is not very useful. So this data might be used for possible treatments in the future. Treatments by focusing on the concentration of different types of transcription factors in the cell, might be focused on looking at how these processes are activated and changing that, or it might be targeting specifically inhibiting TLR4 and IRF treatments before they can make the tumor necrosis factor alpha cause a problem. Through this process, I hope to not only make a discovery, I hope to make a difference with my research. And I'm more than excited to see where this is going to take me. Thank you.
All right, thank you, Rollin. So at this point,